Students, we have studied about spermatogenesis, formation of the sperm. The last step was the transformation of the spermatid into a functional sperm, and that was particularly known as spermiogenesis. Now today we are going to study the structure of the human sperm. Structure of human sperm. This is our topic for today. Sperms are motile cells. They are microscopic structures, and they remain alive in the female genital tract for 24 to 48 hours. A typical sperm, we are talking about the human sperm, consists of 4 parts. One is the head. This is the head. The head contains at the anterior side, towards the anterior side, a structure called the acrosome. This is the acrosome. It is a cap-like structure. And towards the posterior, there is one posterior, there is nucleus. This is the nucleus. Elongated nucleus. It is not spherical. Yesterday I told you, spermatids, when they undergo transformation to form the sperm, there are various changes which occur in the spermatid to form the sperm. This is called the head. This is called the nucleus. The nucleus contains the chromatin material, that is the DNA. And this is the acrosome. Acrosome contains sperm lysins. These are the proteolytic enzymes which will break the wall of the egg at the time of fertilization, at the time of entry of the sperm inside the egg. Below the nucleus, there is present a short, this is known as the neck. The neck contains 2 centrioles. One lies just below the nucleus, and the other lies behind the proximal centriole. The proximal centriole is responsible for the first cleavage of the zygote, and this distal centriole forms this axial filament of the sperm. This is the axial filament of the sperm. This is known as axial filament. Let us label it. This is the proximal centriole, and this is the distal centriole. The proximal centriole like is a regular centriole. This has got 9 plus 2, 9 plus 0 pattern of the microtubules. And this also has 9 plus 0 pattern of the microtubules. This axial, around this axial filament, there are present mitochondria. These mitochondria are arranged in a spiral fashion, like this. Like this, in a spiral fashion. Like this. There are 15 to 20 mitochondria. They are arranged in the form of a spiral. And this is called mitochondrial spiral. So that the mitochondria occupy less space. Then this is called the, let us say, middle piece of the sperm. This is the middle piece of the sperm. Middle piece. Middle piece contains axial filament formed by the distal centriole, and mitochondria are arranged in a spiral fashion around the axial filament, and that is called the mitochondrial spiral. Then this axial filament that continues in the tail region, like this, like this, like this. Tail is very long as compared to the head. This. This is known as the, let us say, axial filament. Axial filament. Axial filament is present in the tail, that is the flagellum of the tail, and it is present in the middle piece also. Because sperm is a cell, definitely it is covered over by a plasma membrane. This is the plasma membrane. This is the plasma membrane all over. And this, around the axial filament, there is present a thin sheath of cytoplasm. This thin sheath of cytoplasm. Let us say this. Thin sheath of cytoplasm. The cytoplasm is present in the middle piece also, because maximum of the cytoplasm has been lost when spermatid was transformed into a sperm. This is again the plasma membrane of the tail region. This is the plasma membrane, you can see. This is the, let us label it on this side. This is the plasma membrane, and this one is the sheath of cytoplasm. Now remember, this sheath of cytoplasm or the plasma membrane does not cover the tail all over. It leaves at one end only the axial filament. The axial filament is naked at the end of the tail. This is called the end tail or terminal tail. We can use the word terminal tail. And this is the sheath of the cytoplasm, or the this is the tail, and this is flagellum. The axial filament, distal centriole, has 9+0 pattern. Proximal centriole also has 9+0 pattern of the microtubules, but the axial filament has 9+2 pattern of the microtubules. Now, what we can say, that this is the number 1 part of the sperm, this is number 2, this is number 3, and this is number 4. You are seeing in the diagram that head, first part, neck is the second part, third part is the middle piece, and fourth is the tail. Now we write the important points. This is the structure of human sperm. This is head, neck, middle piece, tail or flagellum. Then this is 9+0 pattern, this is 9+0 pattern, this 9+2 pattern. Now we come to the salient features or the important points. Number 1. Human sperms were first seen by A.V. Leeuwenhoek in 1677. They were first seen and sketched by, the diagram of the human sperm was made by A.V. Leeuwenhoek. Seen and sketched, definitely under the microscope. These sperms are microscopic, motile cells. Second point. Third, sperms remain alive in the female genital tract for about 24 to 48 hours. Alive. Then they die. Then fourth, about 200 to 300 million of sperms are discharged in one ejaculation. One ejaculation. human sperm, as we are seeing into the diagram, is divided into 4 parts. Number 1 is head. Head is occupied by 2 structures, acrosome, anterior, and nucleus is posterior. Now, this nucleus has chromatin material. Chromatin material. Which is mainly the DNA. Now, very important point. This DNA does not contain any histone proteins. No histones or histone proteins. These are basic proteins. In eukaryotic cells, the DNA is wrapped by the histone proteins. These are basic proteins, very, very, very important question. But in case of the nucleus or the DNA of the human sperm, no histone proteins, but another type of basic proteins, but protamines present. Histones are made up of arginine and lysine. You know it already, we have studied in 11th standard. These protamines are arginine rich proteins. They are also basic proteins. Basic proteins. Again, instead of histone proteins, the DNA contains the protamines. This is a very, very important question for you. These are protamines. And this is the genetic material which we call it the chromatin. Chromatin. It is actually DNA. One thing more about the nucleus, very, very important question for you. This is chromatin material. Second and third, nucleolus is absent. No nucleolus is present in the nucleus of the human sperm. Nucleolus absent. Again, this chromatin material is mainly DNA, no histone proteins, but protamines are present. Protamines are the basic proteins and nucleolus is absent. Now we come to the acrosome. Students, you can note down all these points. All these points. Now we talk about the acrosome. It is cap-like structure, cap-like. This acrosome is formed by Golgi bodies of spermatid formed by the Golgi bodies of spermatid. Third, it contains proteolytic enzymes, and those enzymes are called sperm lysins. Sperm lysins. Lysins means lysis. They will break the membranes of the egg at the time of penetration or at the time of entry of the head in the head of the sperm in the egg. Sperm lysins, one of the sperm lysins, there are so many sperm lysins, that is, for example, hyaluronidase. It is a sperm lysin. It means what we can say, that acrosome acts as lysosome of sperm. Lysosome of sperm. It's a very important question. Lysosome of the sperm. Cap-like, formed by Golgi bodies, contains proteolytic enzymes called sperm lysins. These sperm lysins, we can write it, they break the membrane of egg for the entry of sperm head, because only proximal centriole and head, they enter the egg. Egg for the entry of sperm head and proximal centriole at the time of fertilization. At the time of fertilization, it will break. Now we come to the neck region. One is head, second is the neck region. Second is neck. As seen in the diagram, it is very short part of the sperm. It lies below the head. Some books write neck is a part of the head. So let us discuss its neck. Neck contains two centrioles. One is called the proximal centriole. Second is the distal centriole. Distal means away. Proximal centriole, both the centrioles, they have got 9+0 pattern of the microtubules. Distal centriole, 9+0 pattern. This proximal centriole is responsible first cleavage of zygote. First cleavage. Zygote is a diploid cell. It will undergo mitosis. Cleavage is nothing but it is mitosis. The first mitotic division of the zygote for the development of the embryo, that is taken over by the proximal centriole. This proximal centriole lies just below the nucleus, just lies below. There is a depression that lies over there, that is a proximal centriole. And this distal centriole, it forms the axoneme, or we call it axial filament of flagellum of the sperm. Remember one thing I just mentioned earlier also, that at the time of fertilization, this only the head and the proximal centriole enter the egg. All other parts, starting from here, they degenerate and they remain outside only. I can write it over here. Only head and proximal centriole enter the egg. Only head of what? Only head of nucleus. Head of nucleus and proximal centriole enter the egg. And then this head region will give its chromatin material to the nucleus of the female gamete. Very, very important question for you. Very, very important. And this, I've told you about the distal centriole. Now we come to the third part, that is the middle piece. Number 3 is the middle piece. Middle piece. The middle piece contains mitochondria. Mitochondria, which are around 15 to 20 in number. These mitochondria are arranged in a spiral fashion, in a spiral way. And this is called mitochondrial spiral. Mitochondrial spiral. Now this mitochondrial spiral, because these mitochondria, what is the function of mitochondria? They will provide energy, ATP. For what? For the movement of the sperm. Therefore, they are also called powerhouse or energy house of sperm. These mitochondria are arranged in a spiral way. One thing more, around the axial filament, which was, or axoneme, which was formed by the distal centriole. And this particular arrangement of the mitochondria around the axial filament is called Nebenкern. We can write it over here. This is called, this one is called Nebenкern. I mentioned it yesterday also, Nebenкern. It is the mitochondrial spiral or mitochondria coiled around the axial filament. Then the fourth part is the tail, or we use the word flagellum. It contains the axoneme. Tail can be divided in two parts. Main part and the end part or terminal part. Terminal or end part. The main part is covered by cytoplasmic sheath. This one, this is the main part. We can write it over here. This is the main part of tail. It is covered over by cytoplasmic sheath. The terminal or the end part is naked. No cytoplasmic or plasma membrane. This is the important point, naked, and this is the end part of the tail or terminal part of the tail. The main part is covered over by cytoplasmic sheath and definitely plasma membrane. Now at the end, I explained it earlier also that what is Ncert. The cytoplasmic sheath, which is formed in the middle piece around the mitochondrial spiral, that is known as Ncert. Some books write it that this cytoplasmic sheath is also present in the neck region and half of the head region. But I agree to this aspect that this cytoplasmic sheath is present around the mitochondrial spiral in the middle piece, that is called Ncert. Cytoplasmic sheath around the mitochondrial spiral in middle piece of the sperm. Very important question, Ncert. And this we talked about the Nebenкern, that is also very important part. This is Nebenкern. Now this is the cytoplasmic sheath around the mitochondrial spiral in the middle piece of the sperm. That's all. Thank you very much.